Hi guys, so today we're going to be talking about the cell structure. First thing that we're going to be covering is introduction to the cell, plant, animal, and virus cell, prokaryotic and eukaryotic cell, microscope, organization of the cell, resolution, and its magnification. Alright, so for the introduction of the cell membrane are thin membranes surrounding the cell for exchange between cell and the environment. Cell membranes are partially permeable as trafficking of materials in both directions takes place. Alright, so this image shows you the animal cell, plant cell, and the virus particle. So you may pause this video just to read up the functions of each element. To summarize this, the nucleus contains nuclear envelope, chromatin, and nucleolus, while the cytoskeleton has the microtubules, centrosome, intermediate filaments, and microfilaments. The animal cells and the plant cell consist of plasma membrane, and they also have perisomes, the endoplasmic reticulum, which are rough and smooth. They both have a vacuole, mitochondria, cytoplasm, Golgi apparatus, and lysosome. As for the plant cell, there is the plasmodesmata, which is a channel that connects two plant cells. They have the cell wall, which is the most important differences between animal and plant cell. They have the cytoplasm, central vacuole, cytoskeleton, chloroplast, plastid, peroxisome, mitochondria, Golgi apparatus, ribosome, nucleus, and the endoplasmic reticulum. Whereas for the virus particle, it's a very simple particle which only consists of the capsid, the protein molecules, and the gene which is DNA and RNA. To make it easier to view the differences and similarities between the plant and animal cell, here shows the Venn diagram. So plant consists of cell wall, large vacuole, and chloroplast, while animal has no cell wall, has a centriole, no chloroplast, and small or no vacuole. Whereas the similarity between the plant and animal cells is that they have a nucleus, cell membrane, cytoplasm, mitochondria, Golgi apparatus, ribosome, smooth and rough ER. Alright, so about the virus cell. Virus cells have no cell structure. They are self-replicating DNA or RNA as genetic code. They have a protective coat of protein molecules which are called as capsid. The size of them are 20 to 300 nm. And the virus DNA or RNA synthesizes protein which is with its machinery to make new virus particle. And the important thing that we also need to know is the role of ATP, which is an energy form derived from the mitochondria. So the role of ATP is to carry out Krebs cycle for cellular respiration, carry out oxidation to break down the fatty acid and amino acid to produce energy, carry out oxidation phosphorylation to produce ATP from ADP, and it also contains DNA to synthesize protein. Alright, so for prokaryotic and eukaryotic cells are as small as 0.5 to 5 micrometer. The DNA is circular and they are free in the cytoplasm. They have naked DNA and small ribosome and also no ER. Prokaryotic, they have no compartmentalization of their cell and the cell wall is present. For eukaryotic cell, the diameter of the cell is 40 micrometer. The DNA is contained in the nucleus, DNA in the chromosome, larger ribosome, ER is present, extensive compartmentalization, cell wall present in the fungi or plants. So prokaryotic cells usually for virus, bacteria, something like that. Eukaryotic is mostly for animal, plants, and so on. Alright, so the image shows a prokaryotic cells. The structure that are always present in all types of prokaryotic cells are cell wall, cell surface membrane, cytoplasm, circular DNA, and ribosome. However, the structures that are not very abundant in many prokaryotic cells are the flagellum, capsule, infolding of cell membrane, plasmid, and the pili. The table beside shows the summarized version of the prokaryotic and eukaryotic cells. Alright, next is the microscope. So we have two types of microscope. One is light microscope and the other one is called the electron microscope. For a light microscope, we have two types, which is compound microscope and phase contrast microscope. The compound one is used to examine small samples. Phase contrast is used to examine living cells, and the image appears as black and white. However, for electron microscope, we have two as well. Transmission electron microscope called TEM and scanning electron microscope called SEM. TEM is to visualize the intracellular structures, and SEM is to obtain the 3D view of the sample. So depending on your type of sample or how you want to evaluate them, you can use this microscope depending on its function. Alright, so the two images here shows a light microscope and an electron microscope. For a light microscope, you can see the pathway of light goes from down and you'll be looking from the eyepiece. So it consists of an eyepiece, light beam, objective lens, condenser lens, and the condenser iris diaphragm. So you have to place the sample on the glass slide, on the light, and you can observe it from the eyepiece. For the electron microscope, it works in a way that the electron will be going in an opposite direction of the light. So the electron gun will then produce electrons, which will pass through. Inside has a vacuum field. It will pass through the condenser lens, the specimen, objective lens, projector lens, then you'll be able to view it on the screen. So this table shows the comparison between light microscope and electron microscope. So light microscope, the specimens are prepared easily, whereas the electron microscope takes a long procedure to prepare. Light microscope, living material can be examined, movement of cell can be observed, the colors will be displayed, it uses light rays, it is very cheap, the tool is very small and portable, it uses glass lens, but the resolution is poor. However, for electron microscope, you have to use skilled materials. No movement can be observed due to the cells are dead. The image shown are monochrome. They use electron beam. The tool itself is very expensive and also very large, and it uses electromagnet lens, but it provides high resolution. So same as this, depending on which type of samples you would like to observe, you would have to use the microscope accordingly. As for the magnification, the definition is an apparent enlargement of an object. So to calculate the magnification, it is equals to the observed size of the image divided by the actual size. However, for resolution is the ability to distinguish between two objects that are very close together. The higher the resolution of an image, the greater details it can be seen. So for resolution, the amount of detail can be seen. Hence, if the resolution is higher, the detail are more. So for example, light microscope is only, the maximum resolution is only 200 mm. That means if the object and another point is closer than 200 mm, they cannot be observed. So as seen here, the magnification is the observed size of an image divided by the actual size, or m equals to I over a. So if you see the image beside, if the question was to determine the size of cell P, you would have to first measure using your ruler. Depending on if you're using cm or mm, you have to convert them into micrometer. So once you have already converted them into micrometer, you use the formula m equals to I over a. For this, if they use the example, the question give the actual size is 80 micrometer, so it will be whatever that you calculated in the ruler divided by 80 micrometer, and you will get the answer. So the answer must always have a multiplication sign in front to show that this image has a magnification of times example 700. Last but not the least is the organization of the cell. This is how from a cell it develops into an organism, a whole person. So from cell, it will differentiate into a tissue, more tissue will develop into an organ, all the organs combined will be an organ system, and that's how you form an organism. So for tissue, example of tissue are like epithelial tissue, connective tissue, muscular tissue, and nervous tissue. As for organs, very straightforward, stomach, liver, heart, and all of that. For organ system is called the digestive system, blood circulatory system, and the nervous system. Alright, that's it for cell structure. So these are the important things that you will need to know for cell structure. Don't forget to download the notes if you would like. For anything, you may email to me with all my details below. Thank you.